All right, so let's talk a bit more about oxidation reduction in terms of something most of you probably find more interesting, which is cells. So when we look at cells, oxidation reduction is incredibly important. It is how we process the fuel in our body to extract in energy forms that we can actually use. That's how we break down nutrients. We do oxidations and reductions. Usually we do oxidations to break them down to form energy. We do reductions to make other stuff. Part of that would be energy molecules like fats. Okay? So in the body, the process of combustion actually occurs through a chemical pathway that transfers the energy as it goes in small increments. That way you never have a large release of energy, which would be combustion, right? Does it seem like a very good idea to have little fire generators, combustion generators in each cell? Would that be a safe state to be in? No, that would be really problematic, right? You'd actually have a tremendously difficult time managing your heat. Okay? Does anybody know, by the way, are humans more susceptible to heat or cold? Yeah, so who here thinks heat? Don't be embarrassed to say it, because if you listen to the news, that's what you're going to think, right? You hear about heat deaths every year. They make a big deal out of it. Who here thinks cold? Okay, a bit more. Okay. Cold is the correct answer. It's 5 times the number of deaths every year, right? And that has been for the entire history of humanity. If we make the planet a little bit warmer, we'll actually have less deaths overall. Okay? That doesn't account for things like we're going to flood out coastlines or stuff, though. So, actually, that's very unlikely to cause deaths directly. But in terms of heat deaths, humans actually are very resistant against heat. In fact, there's an experiment that was done by a magician in the 1800s. He put himself in an oven at 400 degrees Fahrenheit. Yes, a fully baking oven. And then he gave himself a tube with a tank of water on the other side and just kept drinking water. And he lasted for 3 days.
Did he get really tan?
No, you don't tan from that, actually. Yeah. The reality is that we will continue sweating. And he actually had to exit because he wasn't getting enough nutrients and salts. So he got very weak over the course of the experiment. But you're very resistant against heat. The planet could be about 4 times the heat it is right now, the temperature, and we would still be able to survive it as long as we could get water continuously. Okay? So it's we are super resistant to heat. Now that doesn't mean everything is resistant to heat. There's a lot of animals that are not. It's just because we have sweat glands, we're quite resistant.
Would we get sick less often since higher heat?
No, we'd probably get sick about the same. The reality is you don't get sick because it's cold. You get sick because you're spending time around a lot of people because it's cold. Yeah, and you're not getting as much sunlight and your other patterns are changing, and so you get different exposures. Yeah. In fact, in terms of sickness, likelihood to get sick, you going to new places is the biggest deal. Have you ever heard of the Paris flu? Or the cruise flu? Okay, there's like these different fun little names for essentially travel sicknesses that you go someplace new, you get exposed to new viruses, you get sick from one. Yeah. And so people tend to get sick on the trips or right after trips. Okay. So the series of reactions through which glucose is combusted, this is actually a type of cellular respiration. And so what's occurring right here is we're using oxygen, we're using glucose, we're breaking the glucose down. That's going to eventually provide us energy. Now the way we actually do this through our bodies is a bit complicated. There's actually a series of steps. Has anybody ever heard of the citric acid cycle? Glycolysis, citric acid cycle, electron transport chain. Okay, I'm seeing some yeses and some nos. That's okay. Okay, that's actually the chain of reactions that takes, those are pathways, each of them are pathways. And so they're about 10-ish steps long on average, a little bit under that. And you would use that 10 steps in the reaction pathways to extract energy by reducing over and over and over the glucose, oxidizing over and over and over the glucose. You would then take that energy, you'd put it in to the citric acid cycle. Again, you would oxidize down the remaining compounds. You would then build up a charge of protons on one side of your mitochondria. Anybody ever heard of mitochondria? Okay. All right. So you build up a bunch of protons, a lot like how a lithium-ion battery works, actually. And that gradient where you've built up protons is actually going to drive what looks exactly like a steam generator. Okay? Everybody seen a steam generator before? Has essentially a propeller that you drive, and then that propeller generates energy and electricity for the outlets. But in humans, that's actually going to synthesize ATP. And it works almost exactly like a press does. Anybody ever seen a joiner or a presser where it takes two items, it puts them together, and it literally just goes thunk? No? It's how they do like the caps on the bottles for the glass bottles. Okay? Or they do like a lot of the different metal joints. They literally take them together and just and smash them together. Has anybody seen that where it has like the brooms usually have them, where it has essentially sheet metal around another sheet metal and it's been pressed together? It's called cinched. Yeah. So that works almost exactly like that. It looks exactly like that. If you watch a video of it and then you watch how it's made next to each other, you'd be like, yeah, that's pretty much that. All right. So ethanol is going to be oxidized, for example, aldehyde, which is ethanol. This is also known as acetaldehyde. By the liver, it's the enzyme that does this is actually alcohol dehydrogenase. In the case of the ethanol oxidation, the molecule is going to undergo reduction as nicoamide adenine dinucleotide. This is the abbreviated NAD+. Anybody ever heard of NADH or NAD? Okay. They sell it as a health supplement now too. Did anybody see that? No? It's an energy booster health supplement that they started selling it as. It's a very bad idea to take. Okay? Anybody know why it's a very bad idea to take? So this is direct energy to your cells. If your cells take this in, it's essentially just giving them energy. But does anybody know what would happen if you gave it and somebody had cancer? Okay, so if you have cells that are taking in, getting energy from it, and you have cells that take in way more nutrients, get more energy from it, which are going to win? The cancer, right? And so this is like growth booster for cancer. It will take a stage 1 cancer and make it develop to a stage 4 in about 4 times the speed. Okay? And so people started showing up with high stage cancers when they were taking this because it was boosting the speed of the cancer growth by a considerable amount. Anyways, so please be careful about that. NAD plus and flavin adenine dinucleotide, which is FAD. These are probably the 2 most important molecules when we're talking about oxidation reduction reactions. There's also an NADP plus as well. Okay? Most people just sort of ignore the P though, because it functionally ends up being very similar to NAD plus. Okay? For all of you, you probably aren't interested in the differences, so good enough. All right. These right here are your oxidation reduction molecules in your body. Okay? So remember how every single oxidation reduction, if something gets oxidized, what also has to happen? Something else needs to be reduced, doesn't it? Okay? Something's reduced, what has to also happen? Something else needs to be oxidized. So whenever you do an oxidation reduction, you're always using one of these molecules as the second thing. Okay? So you oxidize ethanol. There was an NAD here that was doing the other part, the reduction, right? Okay. Okay. By the way, does anybody know what this would be if it's reduced? That's the oxidized form right there. Typically, it's written as NADH. Okay? Okay. It is not actually correct to write it as NADH, but everybody always does it, so. Damn biologists. Okay. If you don't know, it's Michael over there. Biologist. Does a lot of nature stuff. If you ever want to see turtles, by the way, go to his office. He has a really good collection. Okay. So right here we have an oxidation. This is the ethanol here. You just went out for a night of drinking. Your liver is working hard away with your alcohol dehydrogenase. Anybody ever seen those pills you can take? They'll make it so you don't have a hangover the next morning. Okay. Anybody know what's in them? Alcohol dehydrogenase. They're enzyme. Yeah. So they just give you more enzyme. This is why some people can drink alcohol and have almost no effect. Is because they have extremely efficient form of alcohol dehydrogenase. And if you drink enough over a time period, like people that are alcoholics, functional alcoholics, they will develop more alcohol dehydrogenase in their body. Yeah. And so they can drink more and more and more, though it's not necessarily a good thing. Okay? Anyways, so this right here is actually the reaction pathway. You'll notice what we're doing in oxidation here. And so we're taking something that's in its oxidized state, and we're doing a reduction to it. Okay? And this is why people typically write it as NADH, though more correctly, this is actually NADH2. Okay? And that would be the more correct form. Anybody have any questions? Okay. And so now we're sobering up. Yay. We'll go ahead and move on. All right, so here we have our condensation reaction. So this is going to be where water is produced as two organic molecules are joined. Condensation reactions can occur among functional groups that contain an H and a polar bond, and an OH group, so those alcohols usually, that can be removed to form water. What do you notice about the word there? Condensate. If I said I have condensate on my container, what would that mean? Like water, right? Yeah. So condensation, we're giving up water, aren't we? Okay. This is a very, very, very common polymerization reaction, meaning it makes polymers. And so like when you see plastics, a lot of them are made with condensation reactions. But also your proteins are typically made with condensation reactions. A lot of the reactions that make long chains in your body are made using condensation reactions. Okay? Very, very common way of doing that. Hydrolysis reaction. Hydro is again going to be water, and so lysis means break, right? And so we're using water to break. It's going to be the opposite reaction of a condensation reaction. So here we have a condensation reaction going forward. We have two alcohols. One loses a hydrogen, one loses an OH, and we end up with an ether there, don't we? See that? Okay. Now, if we went the other way, notice again we lose water in the condensation reaction. Now, if we go the other way, that's going to be a hydrolysis. And so we're going to attack with water. We'll add water across and split the two molecules. Okay? Notice that we are using a decomposition reaction this way. This one's a synthesis, right? Those are the class of reactions. Okay. Note that these are incredibly important biological reactions. This is how you do almost all the synthesis and destructions in your bodies. I've actually covered like 90% of the most important reactions already with these. Okay? So these are some of the most important stuff already. So again, very, very common in biochemistry. ATP, for example, is actually hydrolyzed to ADP through a hydrolysis reaction. And so this right here is ATP. It reacts with water, H2O. Hydrolysis then gives you a phosphate and ADP. Okay? Does anybody know how long your ATP takes to run out? Let's say you didn't do anything at all. You just sat there, no energy being expended. Did you know that ATP is unstable? So this right here, this hydrolysis reaction, is actually spontaneous. That's a spontaneous reaction. You have water all around in your body, right? That water is constantly hydrolyzing your ATP. Okay? So how long can you go if you expended no energy whatsoever? How long could you survive and still have ATP? Anybody have any ideas? It's about 48 hours. Okay? So in 48 hours, you have lost the vast majority of your ATP. And you would no longer have enough to actually function. Okay? And so that's the amount of time it actually would take you to essentially starve to death if you don't have any fat stores. Okay? Okay, and so this is like that. Notice that that's where you start feeling really, really like the hard hit if you ever do any sort of fasting. Has anybody ever done a fast? Yeah. Like the first 24 hours is The next 24 hours is much worse. And then after 48 hours, it starts getting really bad, right? And then the third, fourth days, it gets better. Okay? If anybody does a full fasting for that long. By the way, that's actually really bad for you unless you take salt supplements. So you need to do a very, very careful fasting procedure. And then it's actually quite good for you, interestingly enough. Okay. So, carboxylation. These are going to be reactions that involve the addition of a carboxyl or carboxylic acid group. This is going to be a carbon dioxide that moves through the cell, and it's been added to and removed from small molecules by two enzymes. These are typically called carboxylase and decarboxylase. So right here, we have bicarbonate reacting with pyruvate. This is going to use up energy in the form of ATP to create oxaloacetate. And notice that we have lost H2O from this. Okay? And so we've added a CO2. See that? We've added a carboxylate. So that's carboxylation, right? If we were to do the reverse, we would remove a carboxylate, right? Bless you.
Thank you.
And so that would be decarboxylase that's doing it. That would be the enzyme. And that would be a decarboxylation. Okay. Note that this right here, if you look at these enzyme names, if you ever want to go into deeper level of understanding of the medical industry or biochemistry in general, then these namings are all named exactly what they do. Okay? So for example, we look here, it's working on pyruvate, that's called the substrate. This right here, carboxylase, carboxylation, it's an enzyme. Okay. All right, so this one right here is going to be the addition or removal of phosphate. This is how you represent a free independent phosphate. That's PI. Everybody see that? Okay. And so this is going to be one way where you regulate your chemical pathways in a cell. The reactions that do this are going to be called phosphorylation. Guess what you do there? Add a phosphate, right? Dephosphorylation. Remove a phosphate. Okay? I know. Chemists were involved in this, so they're named exactly what they do. If it was a biologist that did it, it would probably be named after someone, right? So, there you go. The enzymes that catalyze condensation, again, are going to be the phosphorylases. The enzymes that do the hydrolysis in this case are going to be the phosphatases, is what the enzyme is called. And so this right here is going to be what? What's going on here? We're adding water across the structure, right? And it breaks a bond, doesn't it? Okay. And so is this a condensation? No. This is a what? Breaking, so lyse, right? Using water, hydro. So hydrolysis. Okay. It's being done through a, in this case, dephosphorylation. We're removing the phosphate group, right? Okay. And so what this commonly does is this is actually an extremely common way to either activate or deactivate an enzyme and make it function either better or worse. Okay? So if the enzyme's overdoing it, you don't want everything to get consumed and make this thing over here, you phosphorylate. This enzyme over here is overdoing it, is not running fast enough. You need it to go faster, do more. I need more of that enzyme. You might phosphorylate it. Okay? Notice I used the same thing for both ways. I did that on purpose because it really depends on the enzyme which way that goes. Okay? All right. So hydrolyzable and non-hydrolyzable lipids. Hydrolyzable lipids can react in a hydrolysis reaction. So it's going to be very, very similar in form to form similar compounds. Non-hydrolyzable lipids cannot undergo hydrolysis. So the hydrolyzable lipids, these are going to be things like waxes, fats, oils. These are going to contain an ester functional group. That's your ester right there. See the beeswax there? Okay. Okay. So that's hydrolyzable. And so they can undergo that same hydrolyzation reaction and split that up into its component fats. Okay? They were actually formed through condensation, weren't they? Which is just the reverse. Okay, and so this is actually beeswax in this case being split up to palmitic acid. You would also create an alcohol, a very long chain alcohol, 30 carbon chain alcohol. Don't drink that, please. It would be disgusting. Okay. So this is some fats and oils that can be hydrolyzed to their component fatty acids. These, in this case, actually produce glycerol or glyceride. Same compound, two different names. Okay? And so they also produce then the glycerol that has 3 alcohol functional groups here. These are the 3 functional group alcohols that allow it to bond 3 fatty acids. And this right here is the 3 fatty acid chains that you would get out of this. Okay? A lot of the fats or oils that you absorb into your body are triglycerides or diglycerides. You then split them up. You let them absorb through your stomach lining or intestinal linings, and then you put them back together on the other side. Okay. All right, so the addition of an alkene. So an atom or group of atoms that's added to a double bond in an organic molecule is going to be called an addition reaction. You're adding across an alkene. So the second bond is in the double bond is going to be broken. And you're going to replace that with 2 single bonds to 2 things that attach to this, or 1 thing that attaches to this, in which case it's epoxide. Anybody ever heard of epoxides? Epoxy? Yeah? Okay. So atoms are going to be added across the double bond here. This is an example of an addition. X and Y just are any 2 atoms that can do this. Okay? This could be HBr, but this could be Br2 as well. Okay? Cl2, HCl, there's lots of different ones. You'll note that we've already done an addition reaction and talked about that. It was hydrogenation, where we have H2, don't we? We just added 2 hydrogens across this. Okay? And so that was an addition reaction, and this is the addition reactions. All right, so hydrogenation is probably the most commonly used of the addition reactions, or at least used in the most amounts, if you will. So during a hydrogenation reaction, 2 hydrogen atoms will be added across that double bond, converting the alkene to a alkane single bond. Your catalysts are going to need to be platinum, nickel, or palladium. There's several other ones that have been developed, but those are the most common and the most used. Anybody else look at this and go, my God, platinum? Platinum's used? Seems expensive. Okay. Remember, though, that catalysts do not get consumed in the reaction. So oftentimes they actually do use platinum instead of nickel because platinum's more resistant to being oxidized by water. Okay? And so it doesn't get rusted up and stop functioning. Okay. Okay. And so the reaction is going to be hard to control. Some of your double bonds will reform in more energetically stable trans forms. And so instead of having your cis, you end up with trans fatty acids in there. This results in compounds, again, known as trans fats. You've probably all heard of those before, right? Okay, you have saturated fats, you have trans fats, and you have cis fats. Okay? This reaction is called partial hydrogenation. You can actually do this on purpose if you want by poisoning your catalyst and causing the catalyst not to function as well. Okay? But in a system where they're doing a lot of this reaction, your catalyst oftentimes is going to get poisoned. It's going to get messed up. Okay? You know how the catalytic converter stops working after a time? It shouldn't stop working, right? But what happens to it is other reactions can react your catalyst. Stuff that you're not meaning to happen in there. Okay? And so over time, especially, your catalyst starts becoming less useful, and you don't always get the perfect reaction conditions. You get partial hydrogenations, and you get some other hydrogenation going backwards, and you start getting the trans fats instead of a completely saturated fat. And so here example is these are the naturally occurring cis bonds right here. Notice they make that nice little sort of puckered structure, right? Okay. And so if we look over here, this right here was partially hydrogenated. And what we notice is it broke the bond and then it reformed. And now it's the trans form. Okay? And that's going to create this structure here. All right. Now these have very, very different three-dimensional structures. Okay? So for examples, does everybody see this cis here? Okay, so if I go like this, what's going to happen to the stuff connecting through this? It's going to bend, isn't it? It's not straight anymore, is it? Okay. Have you guys gotten in what cell membranes are? Anybody ever taught you anything about that? Okay. So cell membranes are these fatty acids where the polar heads, which are up here, right, are pointing outwards. And the tails here are pointing inwards towards each other. And so you get a double lipid membrane, don't you? And that's a cell membrane. Okay? Cell membranes have to be very, very carefully controlled in terms of their fluidity, how rigid and flexible they are. Okay? Too rigid, they start breaking up. Too flexible, they also start breaking up and they start letting things through they should not. Okay? And so you very carefully have to maintain the correct order of flexibility and rigidity. What happens if you get a lot of stuff that makes it really rigid incorporated into your cell membranes because you're eating so much of that stuff? Anybody know? Cells get real rigid, they start popping open and breaking, they can't let things through they normally would, and they end up getting stuck places they should not. Okay? So your veins start doing micro leaks and breaks. Does anybody know when you have a lot of damage to your system and your body's trying to repair it, what it does? It ramps up cholesterol because cholesterol is necessary to make your cell membranes, actually. It's part of the process of getting the correct rigidity. Okay? And so what happens when you have heart disease? Your cholesterol levels go up. Is that what caused the heart disease? No, it's not what caused the heart disease. Actually, a better indicator of heart disease likelihood is sugar levels, how much sugar you consume, interestingly enough. Okay, anyways, That's partial hydrogenation. You end up getting trans fats, and then what would have normally been a fairly benign saturated fat is now a trans fat, and you may end up getting too much of that. Okay? And it's not particularly useful for you. Hydration. So hydration is going to be the addition of water to the double bond in an alkene. The water gets added as H and OH. This reaction is often going to require a catalyst, typically something like an acid, or you can use an enzyme. Humans tend to use acids to do this reaction. Obviously, biological uses enzymes because it's difficult to have an acidic environment and still survive. Okay. But some organisms actually have a slightly acidic environment in certain places, so it's kind of an interesting thing. Anyways, this is the addition of that water across it. Again, that's the hydration. This is different than hydrogenation, right? Hydrogenation, hydrogen only. Hydration, water. Okay. Okay. Again, this is the hydration right here. The alkene double bond is asymmetric. If it is asymmetric, then the hydrogen will usually bond with the carbon that has the more hydrogen atoms, and that will give you your major product. That will be the product you get the most of at the end of your reaction. Okay? Whenever you do reactions that have asymmetry, where you put something in, it's not the same on both sides, you are going to get what's called a major and minor product. Okay? Major product is what you get the most of. That could be 99.9%, almost all of it. That could be 100% under these conditions because I can't detect the other one. That could also be 61%, 51%. Okay? You just get the most of that one. Minor products are going to be what you get less of. Okay? It may be essentially nothing. It may be I get 40-something%. Okay? And they're pretty close. All right, so the OH is going to bond to the carbon with more carbon groups. This is actually referred to as Markovnikov's rule, and that was actually the first chemist that noticed this. This was actually a Russian chemist, Vladimir Markovnikov. And so we actually see an example right here where we look at the two sides, and we see that this side has the more hydrogens, this side has the less hydrogens, right? And so the side that gets the OH is the side with the less hydrogens, isn't it? Right there. The side that gets the hydrogen and the hydration is the side with more hydrogens. Okay. By the way, what do you think anti-Markovnikov would be? The opposite, right? So anti-Markovnikov would just be flipping the sides that the OH goes on. Okay, so this is your study guide of what we covered in today's, this Chapter 5 lectures. Does anybody have any questions on Chapter 5 until I go ahead and go on Chapter 6? No questions? Okay, so make sure you practice. There's homeworks. Did I move the homework to an acceptable date now? Okay, fantastic. Please do let me know, by the way. So I have 6 courses that all have different homework dates, different everythings, right? That's not including double classes, none of that. So I have 3 labs for this. That's just counting those as 1, okay? So having said that, because I have 6 of these that I have different assignments, different homework dates, everything, sometimes I don't realize one of them I left out of date. I do the other 5, and I'm like, forgot the 6, right? Okay, so let me know if there's something that you think might be out of date, okay? Okay. Let's go to Chapter 6.